All right, welcome back, zoologists. We are now going to start covering the Amniota. So that's going to include all kinds of familiar taxa like crocodilians and snakes and lizards and turtles and a few other things that maybe you're not familiar with. So we'll talk about the group and the major evolutionary innovations that they exhibit. And then we'll talk about some of the major groups within the Amniota. So we'll talk about turtles, we'll talk about lizards and snakes and tuataras. We'll talk about crocodilians, and later on we'll cover birds and mammals. So let's dive in. So this is a phylogeny from a previous lecture, and the Amniota is right here. So remember, we've already covered the tetrapods, the tetrapoda. This week in lecture, we're going to talk about the Lissamphibia. We, of course, covered all the fish. So now we're going to go into this group. So what do all of the amniotes have in common? So this is a phylogeny from your book, by the way, that shows all the included taxa. And I'm going to walk you through this phylogeny. So don't panic at this point yet and go, oh my God, look at all that crap. What does that mean? Don't worry, we're going to walk through it. All right, so what do all of these animals have in common? Well, the biggest and most important feature is the amniote egg. So the evolution of this was a major innovation because it forever severed the tie that previous taxa had with water. So they were dependent upon water for at least part, if not all, of their life or life cycle. But now the amniotes could separate and step away from the water, and it allowed them a lot of freedom, and it allowed them the ability to invade habitats that weren't really available to them previously. So what is it about this egg that's special that allowed them to do that? Well, there's four major parts of the amniote egg. So the first is the amnion. That's this part here. This is the water sac that completely surrounds the embryo growing inside, and it protects the embryo from injury. Sorry. So it's basically a watery sort of cushion all the way around the embryo. The second is the allantois. So in this image, it's this little red sac here, and you can trace it. If you look to where it's going right here in between the eggs. So it's attaching to the cloaca, or the cloaca. So this is the waste sac. This is where all of the metabolic waste and solid waste are being dumped into prior to hatching. The yolk sac is indicated here in yellow, and you can see it here, and notice it attaches right about where the stomach would be. So this is what's holding all of the nutrients for the growing embryo. So this is where it's going to get all of its energy from. And the fourth aspect is the chorion, or the chorion. So it's pointing to this membrane right here, just inside the shelled egg. So this membrane here. So this membrane encloses the entire organism, all of the membranes, all the embryo, everything. It's highly vascularized. And so it's through this membrane that gas exchange can occur. So oxygen can come in, carbon dioxide can leave, and it's also often water permeable as well. So there is some water that can go through and go out. Now, these four membranes are crucial to the developing embryo for its survivorship. And in this particular case, what I'm showing you is a shelled egg. So this is referred to as a cleidoic egg. So cleidoic eggs are a kind of amniote eggs. All amniotes have all four membranes, but they may be rearranged in a different way when you look at the different groups. The cleidoic egg is a shelled egg, and that's the ancestral condition for all amniotes. And most amniotes still lay shelled eggs. It's only later within the mammals and within a few other taxa independently that live birth had evolved. And only in the case of the mammals did it completely change such that eggs were no longer involved at all. And there's one group of mammals that are referred to as the placentals. So just to show you that in the phylogeny, so here's the phylogeny. Pretty much everybody on here lays eggs. And even though there is live birth in a few of the snakes and occasionally in some lizards, when they have live birth, what they really are is what's called ovoviviparous. So viviparity is when you give strictly live birth. Ovo vi, ovo vi, vi, Ovoviviparous organisms, what they do is they have eggs that developed inside and they are shelled. But then those shells, the embryos hatch out of the shells inside the mother, and the shells are either reabsorbed by the mother, or in most taxa, they actually just come out with the birth. And so what happens is the young are then birthed, and it looks like live birth, but in fact they hatched out of a shell inside the mother. In the case of mammals, though, most people think mammals are just placentals like us. So the embryo grows inside. You have an umbilicus that's attached to the placenta inside the mother's womb. But that's not true for all of them. So remember, of course, one of the most ancestral groups of mammals is the duck-billed platypus and a few other taxa that we'll cover. And they, of course, did lay eggs and don't give live birth. And then we'll also talk about the marsupials, which do things a little bit differently. And then we'll talk about the placentals when we get there. So the amniote egg was a huge evolutionary innovation that allowed for the separation from water. And so now you had taxa that could invade the land in a variety of different habitats. And they could either lay their eggs or they could make nests, or in the case of placentals, you could carry them around. But there are some other features that also are characteristic of the amniota. One has to do with, there's 3 different patterns of what are called skull fenestrae. So fenestrae are holes in the head, and you have all kinds of different holes in your head. And what I'm showing you here are the 3 different conditions. So you have the anapsid skull, the diapsid skull, and the synapsid skull. And they each have to do with temporal fenestrae, so holes that are behind the eyes. Now in the case of the anapsid skull, there is no hole behind the orbit. So it lacks that. There's 2 holes in the diapsid condition, so diapsid, so 2. And there's a single hole behind the orbit in the synapsid condition. And over the years, I've noticed that students get really confused about these holes. So remember, the orbit is where your eyeball goes. So to try to help you with that, I'm going to stick some eyes in there. There you go. So no hole in the anapsid, 2 holes in the diapsid, and then 1 hole in the synapsid. And I think, so these holes are very important because they allow for additional muscle attachments to the skulls by virtue of these holes. So they actually go around the holes and then through the bars here. You don't have it here, but along the sides. And what this allows is for much greater force of jaw contraction and jaw movement. So it allows for much greater strength in the jaws and the ability to manipulate your food considerably. It also allows for, suctioning power where that's necessary. So to kind of give you an indication of this, you see it here in this diagram of these skulls. And what these lines are trying to show you are the forces, the physical forces that are acting on the skulls. And so you can not only, of course, open and close your jaw, but there's quite a bit of flexion that's possible in the jaw, particularly when feeding. And I'll show you some videos to exemplify this in lecture. Another innovation in the amniotes is rib ventilation. So this is a picture from your book to kind of highlight lungs. So we didn't talk about this last time when we talked about the amphibians, but many amphibians have lungs as well. Some don't, and we talked about that. Some do. And the way that they get air into and out of their lungs is through gular pumping. So basically, and I'll show you a video of this in class, but basically amphibians will move their throats open and closed or up and down to force air to go in and out of the lungs. In the case of amniotes, we have a diaphragm that does that for us. And I'll show you a video of the diaphragm functioning, but basically the diaphragm is a muscular structure in your torso that, again, moves up and down and allows for the inhalation and exhalation of air. Okay. So also with the amniotes, there is the evolution of thicker, more waterproof skin. So now that you're away from water, you have to have a way to prevent desiccation. So remember that in the amphibians, many of them had very extremely thin skin, and this was allowing for the permeability of water across it to keep them moist, to assist in breathing, and to assist in all their functions. In the case of the amniotes, they have a much thicker epidermis and dermal layer. And then on top of the skin, there are usually modifications to assist with prevention of desiccation. So either you have scales, you might have feathers in the case of birds, or you might have fur in the case of mammals. And only secondarily are there organisms like humans where you've lost hair because there are other less problematic ways for preventing desiccation, like drinking water. By the way, going back to those jaws, the jaws are actually designed for gripping and for crushing prey. And so I'll show you a video in lecture to compare and contrast what the amphibians doing. Remember, amphibians are not amniotes. And to compare them to what the amniotes are doing. So I have a video I'll show you of lizards that are consuming food and manipulating their food with their jaws, which you just don't see in amphibians. So what they'll do is they'll swallow prey whole and just swallow it, and they don't really manipulate or chew their food, whereas amniotes do. They also have a much more efficient circulatory system, and so I think I mentioned this in class that as we move our way through the tetrapods, if you're looking at, for instance, fish, there's a very simplified heart structure where there's only an atrium and a ventricle, and blood goes out to the gills to get oxygenated, goes out to the body, and then basically comes back around. And so it's a very simplified network. But once you start advancing onto land, you have organisms that have modified this heart structure. So initially with amphibians and just a few reptiles, they have a three compartmentalized heart, 2 atria, and 1 ventricle. And what's happening here is deoxygenated blood, which is indicated here in blue, comes to the heart, gets pumped out to the structures to get oxygenated. In this case, it's usually to the lungs. It then comes back to the heart, and then the heart pumps it to the rest of the body. And this is in part because you're involving a number of organisms that are bigger, not always bigger, but most of them are bigger. And so they need to have a muscular structure to facilitate getting that blood all the way to the body and then all the way back to the heart. Now, one consequence of this arrangement, though, as you can see indicated in purple, is that you have a mixture of oxygenated and deoxygenated blood. And so while this is a more muscular advance over what was happening with, say, fish, it does have the added problem of not being fully oxygenated blood that's going out to the body. And so that might compromise certain actions or activities that you might want to be making, or that you might be able to make. However, later what you get with birds and mammals and a few reptiles, crocodilians in particular, is a fully separated heart. So now you have 4 chambers. You have 2 atria and 2 ventricles. Deoxygenated blood will go to the heart, get pumped out to the lungs to get oxygenated. Oxygenated blood comes back to the heart, but it's restricted to just the left side, which then goes out to the body and then comes back around again. So there's more efficient strategies for water conservation as well, in terms of the metabolic physiology in the kidneys. So again, comparing to the amphibians, which again are not amniotes, they have a pretty good kidney structure, but remember, these guys are tied to the water. So as a result of that, their urine comes out as being what's called ammoniotic, meaning it's very dilute nitrogenous waste. Whereas if you go to the complete other extreme, what you have with non-avian reptiles, so with lizards and snakes, for instance, is you get what's called uricotelic metabolic waste. So the urine literally comes out in sort of a solid or maybe even a semi-solid state, and it's incredibly nitrogen rich. So it's almost acidic. So it's called uricotelic. So it has uric acid in it. And you do find organisms that are somewhere in between. So mammals are somewhere in between that, where yes, they have urea, they have nitrogenous waste, and it's maybe more acidic than what frogs have, but not as acidic as what maybe desert-dwelling non-avian reptiles would have. They also have a more complex nervous system, and I mentioned this in lecture 2. So if you take this as an example of an amphibian brain, which is much more advanced than fish brains, you can see that there's now developments of the lobes that have to do with different actions that the brain is controlling. Now these are colored to show you the different regions, but what you can see is that some of the nerves, major nerves, are coming off here, and there's a development of different sections of the brain, but they pale in comparison to what you see in, say, humans. And so this is the dorsal aspect. Wait, yeah, dorsal aspect. This is the ventral aspect. Same here, dorsal, ventral. And notice each of the regions is similarly colored, but it's even much better developed in the mammalian brain. Many of the lobes are much better developed. And you notice these sulci and gyri. So the sulci, that's plural for sulcus, those are the valleys or the grooves in the brain. And the mountains, if you will, the gyri, are the sort of hills that sort of look like, you know, big spaghetti that have been put together. So the more complex and advanced the brains are, the more sulci and gyri that you see in them. And humans exhibit that to a great extreme. Okay, so those are the major feature of all the amniotes. Now what we want to do is go into each of the major groups. So we're going to talk about the mammals, the tuataras, other lizards and snakes. We'll talk about the turtles, and then the archosaura, which are the crocodilians and the birds. So let's start. By the way, I'd like to point out this phylogeny. Notice that here's the ancestral condition, the anapsid skull at the base. And your initial split is between mammals and everything else. And bear in mind that mammals are synapsid taxa, so they just have a single fused hole behind the orbit. Then we have everybody else. Everybody else are diapsids, with the exception of turtles. Turtles are anapsid taxa. And notice too that there's a trichotomy here. So you have lizards, snakes, and tuataras, which form the Lepidosauria. You have the Archosaura over here, and then the turtles are here as well. So that means there's a lack of resolution regarding where the turtles should go. And it's kind of an interesting problem. This is something that's been under study for over 20 years, and it's still not resolved. It turns out the turtles are very, very bizarre organisms. They have some really bizarre morphology. And even when you look at genetic data, DNA data, despite the fact that they've been around for a long time, so they should be really highly evolved from the standpoint of they've been around, there's been a lot of changes over time, their DNA is pretty slowly evolving. It makes it incredibly hard to determine who their closest relatives are. And it's still under study now. Even though lots of people have tackled this with lots of data, it's still not very well understood. So we're actually going to cover them first as the weirdos, and then we'll go and talk about the other taxonomic groups. But do remember, we'll come back to mammals last, but mammals are synapsids, turtles are anapsids, everything else is a diapsid. Okay, so let's do turtles first. And by the way, notice I'm not using any ranks. I'm not talking about orders or classes or anything like that. You're probably going to get some of that in lab, but that seems to be under transition as well right now. I think I mentioned in lecture that there's been this increasing trend towards getting rid of the ranks and just going with the major names for the groups and making indented lists when it comes to classification. And that's again because there aren't enough ranks to actually go with all the taxa that are in our phylogeny, so all the taxa that are on the earth. So I won't be using those. I'm just hoping that you'll remember, or that you should be committing to memory the major names of the major groups. And I'm going to walk you through those right now, and we'll talk about them in class and we'll make lists. So the turtles are first. As my advisor once said years and years ago, any idiot can recognize a turtle. They're just characteristic taxa that are not like anything else. They have shells. They have a top shell and a bottom shell. So they have a carapace and a plastron. Nothing else has a shell like a turtle has. Now here's another representation of that turtle. So if you didn't know, the shell is actually composed of mostly bone, and that bone is composed of fused vertebrae in their backs, and then their ribs are completely fused together. And what that does is it creates a really interesting situation where the girdles, the pectoral and the pelvic girdles, are literally inside their rib cages, which is not like us at all. If you look at a skeleton of a human, you will see that the ribs are on the inside, and your like clavicles, your pectoral girdles, and your pelvic girdles are completely outside your rib cage. With turtles, they're all on the inside, which is kind of weird. There's quite a bit of diversity among turtles worldwide. Oh, don't forget they're anapsids. So we've got all kinds of things like map turtles, which you can see here in Michigan. There's all kinds of tortoises around the world. There's matamatas, which are aquatic turtles. And of course, snappers, which we also have. There's a few other things I'd like to point out about turtles and their morphology and myths. So this image that used to be in your textbook shows the neck out here and then the neck being withdrawn here, making this very curvaceous neck. Just so you know, not all turtles can do that. So there's two major groups of turtles, those that can withdraw their necks like you see here, and sometimes they can even close up their shells. But many turtles around the world cannot do that. They're not physically able to withdraw their necks into their shells. But what they can do is actually turn their necks to the side, and they can actually put it up under the edges of their shell so that they're protected. And that actually forms a monophyletic group referred to as the side neck turtles. And if you want to know more about that, we'll talk about that in herpetology in the fall. So the top shell is referred to as a carapace. The bottom shell is a plastron, and there's a lot of variation among turtles as to how big that plastron is. Sometimes it covers the entire bottom part or ventral part of the turtles. Sometimes it's very small. It's almost like a turtle bikini, which is kind of what you see in a lot of the snapping turtles. Sometimes the shells are hinged on the plastron, which facilitates them being able to close their bodies completely up inside the shells. Most turtles cannot do that. They don't have that adaptation. Generally speaking, not always, but generally speaking, terrestrial turtles that never really go into the water and don't live in the water are referred to as tortoises. And generally speaking, those testudines that are aquatic are referred to as turtles as opposed to tortoises. And I think I neglected to say, so this is all turtles belong to what used to be the order. That rank is changing, but the order Testudines. So some other things you should know about turtles is that they all lay eggs, and they look terribly cute. And they exhibit a really interesting phenomenon that you should commit to memory: temperature-dependent sex determination. So you're more used to the human condition and maybe the mammalian condition where being male or female and having the secondary sexual characteristics associated with those conditions is genetically determined. And with us, it's a chromosomal difference. So you're either XX, which is female, or you're XY, which is male. And there's certain characteristics that are coded for on those chromosomes. But in the case of some reptiles, and specifically here turtles, their sex is determined on temperature. And by that I mean it's the general average temperature at which those eggs are incubated at. Now, as it happens, turtles exhibit some variation in this, and they're showing you two examples here. So this is common snapping turtles, and this is river turtles. And so what you can see here in this graph, so forget about the crocodilians for now. So what you're seeing on this axis is the sex ratio, so the percent male. So at what temperature do you get 100% males, let's say, or majority males? And then at the lower temperatures is when you'd get primarily females. And what you see in some turtles is that if you look at the range of temperatures that on average the eggs were incubated at, and in this case you're looking at something that's around, call it not quite 23, all the way to 30 degrees. In some turtles, the low temps and the extreme high temps will produce females, and the middle temps will produce males. In some turtles, as it happens, the low temps will produce males, and the high temps will produce females. So there is some variation in the turtles. The take-home message I want you to get for now is simply that turtles exhibit temperature-dependent sex determination, and you get different sexes depending on the temperature at which those eggs are incubated. For right now, I'm not concerned about you knowing the exact graph. All right, so now let's talk about the next group. So if we're looking at our phylogeny here, so here was the first split, and we talked about the turtles, the weirdos. So here are the taxa that are included within the Lepidosauria. So there's tuataras, there's lizards, and then there's snakes. And I want to point out something that's completely wrong in this phylogeny, by the way. So in here, they're showing like a couple of branches that are supposed to represent lizards. These are monitor lizards, so things like Komodo dragons are included within the monitor lizards. There's several different species of monitor lizards. And they're showing the monitors as being the sister taxa to the snakes. Now, we now know that that's not the case. It is true that snakes form a monophyletic group within the lizards, but the sister taxon to the snakes is unknown. There's been a lot of data thrown at this in the last 10 years. There are a number of taxa that keep coming up as possibly being sister taxa to the snakes, but we really don't know. So do not commit this to memory. What you want to know is that there's several different branches of lizard groups in here, so things like chameleons and geckos and iguanas and skinks, and so all kinds of groups of lizards. And then near the top are snakes. So snakes do form a monophyletic group that's highly derived within the lizard group. And then you have tuataras. So what do all lizard snakes and tuataras have in common? Just a few characters. One is, if you look at the arrangement of the opening of their cloaca, it's transverse. So it's sort of side to side. So here's some lizards, and you can see they have a transverse cloacal opening. It almost looks like it's smiling. This is in contrast to what you have with, say, amphibians, where in their cloaca, it's a sagittally oriented, not transverse. So the opening is right here. Another feature that they share is that they shed their skin all in one piece, or all at one time anyway. So it looks like it's coming off in patches in this anole lizard, but what's happening is they do shed it all at the same time. It just may break off in pieces as it's coming off the body. Now there are a number of skeletal features that they share in common, but compared to your education at this point, they're a little bit esoteric, so we're not going to go over them. Just suffice it to say that there are several skeletal features that also unite them as a group. So now let's go into the three groups. So we got tuataras, we have snakes, and we have lizards. The tuataras form a monophyletic group. As it happens, there's only one species in the group, and they're in a clade that's referred to as it's a Sphenodonta now. Actually, the name has changed, so pardon me while I change that right here live. So that name has recently changed, Sphenodonta. Let's go back in. And right now there's only one species. There was as many as 3 just a few years ago, but they've been synonymized again. So these, for all intents and purposes, they look like lizards. Like there's no reason if you just saw them out in the field why you'd think that they weren't a lizard. But it turns out that they have some really unique features that separates them from lizards. The first is that they lack hemipenes, and I'll explain what those are in just a moment. But they don't have any kind of intromittent organ or a penis of any kind. So they reproduce like birds do. They just put their cloacas together, they transfer sperm from the males to the females, and then they have internal fertilization of the female's eggs, and then she lays them, and they hatch at some period of time later, a month to 2 months later. These guys are all nocturnal. They're only active at night, not during the day. And they only occur on a few islands that are off the coast of New Zealand. And that's something I want you to commit to memory. So you got Australia. Off the coast of Australia is New Zealand, which is formed of 2 major islands. And then there's a couple of really small islands like Cook Island that's off the coast of New Zealand. And that's where you find these very odd lizards. So now let's talk about the true lizards and snakes. So they're in a clade called squamata, so the squamate, so the kind of low to the ground, creepy crawlies, if you will. So what features do they share in common? What unites all lizards and snakes? Well, they have a very highly kinetic skull, meaning that they can deform their bones quite adeptly, particularly snakes. And you probably already know this to be true. If you've probably heard or seen that snakes can can separate parts of their bones to feed on very large object or items. That's kind of sort of true, and I'll go into that in just a minute. But both of them have the ability to actually quite easily move bones in their head for a variety of purposes. So they're highly kinetic. The other major feature is that all lizards and snakes possess what are called hemipenes. So this is a structure that that males use to inseminate females. This is literally one of them, and every species has two sets. So this is one set of hemipenes. There's another set on the other side, so one on the left and one on the right. Sometimes they're bilobed, as you see here. Sometimes they're unilobed. So here's a snake where you're seeing one set of hemipenes and the other set on the other side. By the way, hemipenis is singular, hemipenes is plural. There's an enormous amount of diversity among hemipenis structures, and they're actually species specific. So you could literally have just the hemipenes laid out on a table, and you would be able to identify each of the species that it belongs to. So you can see some of that diversity here, and these lines right here, this is the sulcus down which the sperm travels as it's traveling towards the female to fertilize the eggs. So unlike a penis, which is basically a hose or a tube, and the hole that the sperm is going to travel down is enclosed, in this case, there's like a groove along the hemipenis structure that the semen travels down. And once again, you can see that sometimes they're single-lobed, sometimes they're bilobed, really deeply bilobed, sometimes it's just a little bit lobed. And what they do is when they reproduce, the males will alternate the use of hemipenes when they're inseminating the females. So they'll use the left-hand side, and then they'll use the right-hand side when they go to inseminate a separate female, and they alternate between. So they fully insert this one to inseminate one female, and then later when they go to mate with another female, they'll insert that one, and they alternate use. So all lizards and snakes have hemipenes in common, and they have a really highly kinetic skull. So now let's talk about the differences between lizards and snakes. So are there any differences? Well, there are characteristics that allow you to distinguish them, but it turns out that while snakes do form a monophyletic group, lizards do not. Okay, and I'll show you a better phylogeny of that in class. But if you were trying to tell the difference between a lizard and a snake, generally speaking, could you do it? Yes. So lizards have movable eyelids. So just like you, they have eyelids that they can open and close. They have external ear openings. You can see them right here. They don't have ears, but they have ear openings. And if you look in there, you can see the tympanum where the sound vibrations are hitting, allowing them to hear. And there is a lot of diversity among lizard taxa. So you have things like geckos and skinks and iguanas. And if you want to know all about that diversity, you can take my herpetology class in the fall. But one of the features I would like to point out is that bear in mind that not all lizards have 4 legs. Most people think, oh, lizards have legs, snakes don't. Not true. So there are several taxa of lizards around the world that have lost their legs, so they're limbless. And we even have some in the state of Michigan as well. So they're known as the glass lizards. So if you were to pick these up, initially you might think it was a snake, but it's not going to feel right. It almost feels like it's armored. The scales are really hard on the outside of the body. And you would be able to see that it's got an eyelid that it can open and close, and it's got an external ear opening. In contrast, when you're looking at the snakes, snakes lack those movable eyelids, and they also lack external ear openings. So you'll never see those ear openings on the sides of their heads. And some people seem to find it very odd and creepy that snakes just have their eyes open all the time. They never actually close their eyes. Now, as it happens, they have a structure over their eye called a spectacle. So it's a special scale, a clear, translucent scale that protects their eyes. It prevents it from desiccating. It prevents, dust and gunk and, getting on it and scratching their lenses. And they shed that whenever they shed the rest of their skin. They are clearly elongated, although some lizards are too, but they have an extreme elongation of their body. And as a result of this elongation, they also have a rearrangement of the organs within their body. So to kind of indicate that, sometimes what's happening is if you have paired organs, they'll be offset in snakes like this laterally. And you can see this, for instance, here with the kidneys. Here's the the left-hand kidney. Here's the right-hand kidney. Same thing with the testes. Here's the left, here's the right. But then in some cases with paired organs, one of them is actually missing to make room for some of the other organs. And you can see that here in the, well, not in the liver, but in the lung. So here's the right lung, let's say right here. So some organs you have one that's lost, or they're just rearranged in the body so that they can fit all in the tube that forms the snake sort of general morphology. So there's a lot of specializations within both lizards and snakes, but to just kind of give you a taste of this, snakes are well known for having all kinds of specializations for eating prey. For instance, there are several snakes that specialize on consuming eggs. And so once they're able to get that egg inside their throats, they have specialized processes on their vertebrae. And what they do is they basically push down into the egg and they basically crack the egg open to release the yolk and the contents. And so then they consume the inside of the egg, but they usually spit out the eggshell itself. They're also clearly quite able to rearrange their jaw structure so that they can put really large objects into their mouths, which they otherwise wouldn't be able to consume. And so they don't necessarily separate the bones in their skull necessarily, but they do unhinge their jaw a little bit. And then there are some membranes that allow them to stretch the bones in their skull so that they can actually open their mouths very, very wide. And if you want to know more about that, we'll talk about that in herpetology. They also have some really interesting other senses that have been developed, but only in some taxa. So there's a group of venomous snakes, the pit vipers, that are well known for having this specialized pit just behind their nares. So this is what where air goes in and out of their lungs. But these structures are incredibly sensitive to temperature, and it's really highly innervated with very sensitive nerve endings. And what it's sensing is the temperature in the outer chamber versus the inner chamber inside the body. And so what this allows them to do is to detect really sensitive changes in temperature, which helps them to track prey or just to track other organisms in their environment. And so I'll show you some videos of this in class, but they can detect temperatures up to 0.01 degree Celsius. And so it's very sensitive at being able to find their prey. And they do have very good eyesight as well. Many people think of these pits as somehow giving them infrared sight. It's not quite like that. That's how it's often depicted. It's not quite like that, but it's similar. So they can actually pick up quite sensitive temperature changes. And of course, in combination with their Jacobson's organs, they can also detect really fine changes in chemosensation as well. So despite what this organ or what this image is showing you, here's the Jacobson's organ, which is directly connected to the brain. And most people know that snakes will, or in some lizards, will stick out their tongues, sense the environment, basically taste the environment, and that the thought is that they then put the ends of their tongue into the Jacobson's organ up through the roof of their mouth, and that's how they sense or taste the environment. As it happens, that's not true. That's not exactly how the Jacobson's organ works, but I'll explain that in class. Okay, so now we've talked about the Lepidosauria, which is all of these guys together. We got the Sphenodonta, and then the Squamata, and then we also have the Serpentes and the Lacertilia are the lizards. Okay, we've talked about the Testudines, and now we're going to talk about this last group, the Archosauria. So this is a group that includes crocodilians, birds, and dinosaurs. So don't forget, dinosaurs are in this group. Okay, now we're not going to talk about the dinosaurs much. And as it happens, there's a new paper that came out a couple of weeks ago that changes the views on dinosaurs. So I'll try to straighten that out in class. But let's talk about this group. Now, one thing that's long been annoying to me and hard to grasp are some of these features that unite crocodilians and birds. And there are a lot of them. But the two main ones that most textbooks point to, and yours does, the first being that the orbit, so the eye hole, is shaped like an upside down triangle. And I have looked and looked and looked for years to find really good images of this, but it seems I'm at a loss to actually find any really good ones. And it seems that this feature is primarily possessed by extinct taxa. There's not that many extant taxa that seem to really display this feature. So for instance, this is a picture that used to be in your book of a dinosaur, and here's the orbit. And if you ask me, this is mostly kind of square. That's what I'd call it. So despite the fact that your book says it's supposed to be an upside down triangle, um, what I'm seeing here is it's kind of square. So you still need to know it. I'm just saying it's confusing, and I'm unlikely to ask you about it. Also, they talk about how all the members in this group possess antorbital fenestra. So there's a hole that's ant in front or interior in front of the orbit. So here's the orbit. Oh, here you go. Here's an antorbital fenestra. And as it happens, this is the nares. This is what goes to the respiratory structures, right? Here you go. Now, another problem with that, though, is if you look at pictures of skulls for extant taxa. So here's a crocodilian, right? Well, here's the orbit. Now, presumably the antorbital fenestra would have been probably right about here, but over time with extant taxa, maybe it's been fused. In addition, this doesn't really look like, where'd my cursor go? It doesn't really look like an upside down triangle for the orbit, but and just as a representative bird skull, here we go. So here's a hawk. So here's the orbit. Now don't worry about this. This is a specialized structure that we'll talk about when we get to birds. But basically, this hole is the orbit. So this is probably the antorbital fenestra in this particular species. So okay, but to me, this still doesn't really look like an upside down triangle. So these characters are problematic. Okay, I'm just going to admit it right there. And so I'm not going to focus too much on these on exams, but when you actually take classes in ornithology, maybe you should, ask about that. But other features that do make sense that are really obvious that you can in fact see clearly are birds and crocs both have a crop and a gizzard, and and they also have a structure called a proventriculus between them. So of course, in the case of birds, they don't have any teeth, right? So they can't really chew up their foods. They might have really good sharp beaks where they can rip off, bits of food, or in the case of seed eaters, they're taking up seeds. But they don't have a way to actually physically break down their food items. And so what happens is often both crocs and birds will sometimes eat pebbles or rocks that they'll then store in their crops, and that helps to physically sort of break up the food before it then goes in for more physical as well as chemical breakdown in the gizzard, and then ultimately it'll go into the gut to be absorbed and used for energy purposes. So the crop and the gizzard, particularly the gizzard, are there to help physically break down the food items for them. Now, as it happens, crocodilians do have teeth. They have teeth in sockets, but they're also using them primarily to grasp prey, not to chew up their food or masticate it, because they're not really built for that. Both of them have a 4-chambered heart, so you can see the right and left atria and ventricles, and notice how muscular these ventricles are, particularly the left ventricles. So I want you to think about why it is the case that the left ventricle is so much more muscular than the right ventricle. Or maybe you might think of it as being like why the right ventricle is less. Whichever way you want to put it, but think about that and I'll ask you in class. However, suffice it to say that there's a full septal separation between the right side and the left side so as to prevent oxygenated blood from mixing with deoxygenated blood. And that makes sense because we're talking about some really, really, really active creatures, birds and crocs. Characteristics they also share, and other characteristics they share in common is parental care. They are both incredibly adept at taking care of their offspring. Crocodilians will take care of their offspring up until about 2 years of age. With birds, it's usually a little bit less, depending on the bird species that you're talking about. But they will spend their time caring for them, keeping them out of danger, feeding them. They're quite sweet. Also, I should also say, should have been on this slide, they also vocalize. Birds are, of course, famous for their singing, but as it happens, crocodilians also vocalize primarily to determine territory and sometimes for mating. It's primarily for between males to determine whose territory is whose. And it primarily consists of growls. It's not quite as complex as bird songs are, but nevertheless, they do both vocalize. So right now, let's finish up by talking about the crocodilia, and then next week we'll talk about the birds. So what do crocodiles have that distinguish them from the birds? Well, one of the features that your book points out is that they lack hemipenes like snakes and lizards have. They have a true penis. But as it happens, when it comes to birds, birds don't have a penis at all. They have the cloacal kiss. They have no intromittent organ. But nevertheless, your book talks about the fact that these lack the hemipenes. They have true penises. They have a complete secondary palate. Now, this skull that I'm showing you here, this is mammalian. It's not a crocodilian. I couldn't find a really good one that shows you the secondary palate. But here's the palatine right here. Okay, basically you have a hardened structure, and you actually have a hole right here that you can go into the sinuses with. So what this allows is for the separation of the respiratory structures from the feeding structures, so that food can actually go into and down your throat, down your esophagus, without going down your trachea and blocking your breathing. So you can breathe and eat at the same time. Okay, without the secondary palate, you couldn't do that. They have dentition, just like mammals and birds do, as it happens. So they have teeth in sockets, although they have no variation among their teeth. The dentition is the same throughout all crocodilians. Here's where I mentioned the vocalizations, and I'll show you a video in class where you can hear the growls. And crocodilians also exhibit temperature-dependent sex determination. So going back to this graph, remember this is percent male. So in the case of the average incubation temperature, low temperatures and higher temperatures will produce females, and it's the mid-range temperatures that seem to produce the males. Okay, we'll finish up next time with birds, and then we only have the mammals after that, and we are done for the semester. So enjoy this, and I'll see you next time.